Okay, so what are we going to do today? We're going to focus on transcription. And we're going to focus it first this process as it occurs in prokaryotes. Okay, and then we're going to contrast and compare the process in eukaryotes. So within transcription, which is basically defined as transcribing or making a copy of. So it's a synthetic process. We're going to look at promoter binding of the RNA polymerase and how it starts transcription, how it elongates the nascent RNA transcripts. So nascent means new or the newly generated, and how it terminates the transcriptional process. And then we'll focus on eukaryotes, which is extensively different than the prokaryotic process in that there's 3 different types of polymerases compared to what we see in prokaryotic transcription. Highly regulated processes of creating these transcripts. And again, there's transcription factors that will regulate this process. And then again, in difference to what we see in prokaryotes, the primary transcripts, in particular mRNA, is extensively modified. So we can talk about adding a 5-methyl G cap, a poly A tail, splicing, all those mechanisms. And then we probably won't get to this today, but we're going to talk about the control or regulation of gene expression in both of these cell systems, okay? So those are the focuses of today. So in prokaryotes, before we start off, just wanted to show this schematic animation of the central dogma of molecular biology. Does anybody know what the central dogma is? Yes. What's that? Yeah, so it's basically how you take DNA and you go to RNA, and then once you've generated that RNA to make protein, okay? So this basically is the central dogma of molecular biology. So we all know that the genetic information contained in this DNA is the blueprint for proteins. So that's the end product. Why are proteins so important? Anybody know? Yeah. Well, don't your cells need proteins to carry out whatever functions they do? So basically, the structure, the diverse secondary and tertiary structure of these proteins is highly critical to cellular survival. So we can talk about different types of proteins if we think about transport proteins. So what's a transport protein in a cell? You probably already talked about in 210. How about hemoglobin? Hemoglobin transports oxygen. Right? You can have structural proteins, so those that are involved in creating the cytoskeleton. You have enzymes. Enzymes carry out enzymatic reactions, okay? And you can also have receptors, so transporters, pumps. All of those are proteins, okay? So the central dogma of molecular biology basically states that this is a unidirectional process, so going from DNA, RNA, to protein. And this was first established by Francis Crick in 1958. But is this true today? How many say yes? How many say no? Yeah. It's not, right? Because with the discovery of reverse transcriptase, you can actually take single-stranded RNA and go back and make double-stranded DNA, okay? Fortunately, there hasn't been any process discovered where you can actually go from protein back to RNA. So this is probably the end product of this linear pathway. And of course, you can't really take the protein structure and go back and predict the DNA structure. And why is that? Yeah. All the different, right, correct. All the different processes that you undergo in synthesizing the DNA to RNA, and then finally to the protein. So you have splicing, you have capping, you have removing of introns, and so on. Really, that sequence won't predict what your DNA was in the beginning, okay? All right, so that's the central dogma of molecular biology. All right, so let's do a comparison here on the structures of RNA polymerase. On the left here, this is the prokaryotic RNA polymerase compared to the eukaryotic. Are these structures similar? Do they look similar to you guys? To me, they kind of do. However, one has 5 subunits in the prokaryotic RNA polymerase. The eukaryotic, you can have between 7 to 12, okay? They carry out the same activity in the catalytic site, but the overall structures, again, are quite different, but they look very, very similar. We talk about RNA synthesis, again, the process of transcribing in 3 stages. So there's an initiation stage, an elongation stage where you just keep on adding ribonucleotides to your nascent chain, and the termination process, okay? So how the RNA polymerase recognizes to stop transcription. Okay, so what are the functions of RNA polymerase? And this is definitely a good slide to know. So I'll just highlight some of the key things that you should remember. One of its functions is to recognize initiation sites, or promoters as they're called. Okay, so these are cis-acting elements typically found on the same strand of the gene that you are transcribing. Okay, so that's what cis stands for. In E. coli, there's about 2,000 promoters that RNA polymerase can find. It also functions to unwind double-stranded DNA. And you need to unwind double-stranded DNA so that you have a template for the RNA polymerase to start transcription. It carries out the process of correctly inserting the ribonucleotide onto the nascent RNA transcript. This process is unidirectional. One RNA polymerase will completely synthesize an entire gene from the start to the finish. And it's processive, okay? So it does it all at once. Termination, it's responsible for recognizing when to stop transcription. And it also can interact with transcription factors that will regulate whether transcription is increased or repressed, okay? And it carries out that fundamental reaction. Does anybody know what the fundamental reaction is of RNA polymerase? What are we doing? It's quite similar to DNA replication. We're just adding nucleotides together to create a what type of bond? Phosphoester bond, right? So they carry out the same fundamental reaction as DNA replication. Okay, so this slide, just a bit of review. These are some of the concepts that you can't forget, even though the final exam is not cumulative. Remember your purines, remember the numbering system. We have adenine and guanine and our pyrimidines. Here we're introducing the new base uracil for RNA. Again, remember we're not going to see T in RNA other than ribosylmetallate if we have a ribose sugar attached to it. Okay, so remember the numbering system and your bases. Another key thing I want you to remember is the glycosidic link that is found within these ribonucleotides. So for the purines, what number nitrogen is this, where the glycosidic link occurs? 9. And then for pyrimidines, it is 1, 2, 3, 4, 5, 1. Yes. Okay, so that's for the pyrimidines, and this is for the purines. Okay, so don't forget your glycosidic link. And again, remember our ribose sugar has that 2' hydroxyl attached to it. All right, so remember structure of the bases and the glycosidic links. Okay, so how do we carry out this fundamental reaction? It's depicted in this cartoon diagram here. Key things to note about RNA synthesis is that transcription always occurs 5' to 3'. So if we look at this diagram, our transcript that's being synthesized starts up here. So we have a 5' end up here, and this is our 3' end. And this happens because the template strand that the polymerase uses is 3' to 5'. Okay? So this is our DNA template strand. So we're growing the chain, and the next ribonucleotide would actually be inserted down here. So we insert our correct base, we get the match, and then this is the fundamental reaction that happens up here. So basically you have that 3' hydroxyl, nucleophilically attack the alpha phosphate on the incoming ribonucleotide. Okay, so remember our phosphates are labeled alpha, beta, gamma in this incoming ribonucleotide. And What's released when we have that reaction happening? What do you see being released? Pyrophosphate, right? The hydrolysis of this pyrophosphate by H2O really drives that reaction forward. So it drives the reaction. Okay? And then we get our phosphoester link, and then we can have an incoming ribonucleotide come in and use that new 3' hydroxyl to nucleophilically attack the alpha phosphate of whatever, not deoxy, NTP that comes in, so ribonucleotide. Okay? And the sequence is complementary to that template strand that we see. Interestingly enough, we talked about how DNA polymerase needs a primer to start synthesizing DNA. Here, RNA polymerase does not require a primer. Does anybody know why, even though we haven't talked about it yet? Why doesn't it need a primer? What is it using, actually, to figure out where to start transcription? It's using those promoter regions, right? So it's kind of like a primer, but you have the promoter regions that are found in the DNA to help the RNA polymerase recognize where transcriptions should start. The RNA polymerase, unfortunately, has no proofreading capability, unlike DNA polymerase. Is that a good thing or a bad thing? You know, it is a bad thing, but it's tolerable. Do you know why? Could possibly suggest why? Yeah. Yeah, so you're going to get lots of transcripts that are being made. So if you have an error in maybe one of them, you know, that's okay, it's tolerable. And we'll also see with the genetic code, if you make a substitution somewhere, most times it still specifies the same codon, the amino acid that's to be incorporated into the protein. So you're kind of okay, even though we don't have that proofreading capability, although, again, not really favored, but it's all right. Remember what Dr. Brown had talked about the error rate in DNA being 1 in 10 to the 10. RNA, it is 1 in 10 to the 5. Okay, so that's a key number I want you to remember. A big difference between DNA polymerase. It also moves much more slowly than DNA polymerase. Dr. Brown had mentioned you can incorporate 800 nucleotides per second. RNA polymerase works much slower at 50 nucleotides per second. Okay, so those are key values that I want you to think about. Okay, so we're going to look at the prokaryotic RNA polymerase in E. coli. I've got some general features in this table about it. It's a very large protein, so if you were to add the masses of these subunits together, it's about 400 kilodaltons. If you forgot what a kilodalton is, have a look at your 210 notes. There's 4 different subunits, okay? So there's an alpha, a beta prime, and a sigma 70 subunit. The sigma 70 is very important because this is what recognizes the promoter regions. Okay? The holoenzyme is actually a pentamer. So even though there's 4 subunits, there is 5 subunits that make up the entire enzyme. And that's because we have 2 alpha together, okay? A beta prime, and then our sigma 70. If you want, when you get rid of that sigma 70, you can call the remaining subunits together the core enzyme. And this becomes important because the role of the sigma 70 is really basically to find those promoter regions, initiate elongation of the transcript for about 2 to 3 nucleotides, and then fall off of that holoenzyme so that you're left with the core enzyme. And that core enzyme contains the catalytic active site. The sigma subunit actually decreases the DNA binding affinity of the holoenzyme because it's essential for you first to find those promoter regions and then start synthesizing some of those ribonucleotides into a chain and then have it fall off. So the core enzyme has higher affinity than the holoenzyme for the DNA. Okay, so once sigma is released, it's about, again, around 8 to 10 nucleotides. and then it can go off and bind to another core enzyme to carry out transcription somewhere else. Okay, you don't have to memorize these numbers again. If there's something important, I will provide it for you. Okay, so just a quick comparison of the active sites where we have that fundamental reaction being carried out. Key thing I want you to note, it's just exactly the same to DNA polymerase in terms of the reaction. So you can think of this as a partial active site representation if we were to draw in our protein chain. So we have some amino acids. They're functional groups that kind of stick out into the active site. There's 3 key aspartic residues that are essential for this catalytic reaction. And these 3 are coordinated to a metal ion, which is typically, which one do you think it is? What metal ion do you think it is? It's actually magnesium. Okay, so you need magnesium for the active site. And it's also coordinated to the growing RNA chain, so the nascent transcript that you're synthesizing. So this would be the 5' end, if we were to extend it up here. And then here's our 3' hydroxyl down here. So it's coordinated to that as well. All right? This is our incoming ribonucleotide. Okay? You'll notice that the incoming ribonucleotide also has a magnesium attached to it. So we need 2 magnesiums in the active site to carry out this catalysis. Okay, one is found in the active site, the other comes in with the incoming ribonucleotide. So once we get the reaction happening, here's our alpha phosphate. We can get nucleophilic attack, the hydroxyl. Once the reaction happens and pyrophosphate comes out, that magnesium ion goes with it. Okay? Right, so again, we've got 3 key aspartic residues. They're essential for the catalytic reaction, all coordinated to magnesium, which also coordinates the growing nascent chain. Our incoming ribonucleotide brings in the other magnesium ion so that we can get the catalytic reaction. So not only is it required here for the coordination, but it also stabilizes the transition state when we carry out this enzymatic reaction. Okay? So you need 2 magnesiums for the active site. Okay, so we briefly mentioned that the RNA polymerase looks for promoter regions. And again, we're going to talk about a couple of the promoter regions that we see in prokaryotes. I first want to explain how these promoter regions were first identified. And they used the powerful technique of footprinting. So this is a pretty creative process that lets you identify binding regions for proteins on DNA. And it's a very simple procedure, so we're just going to walk through it. Typically involves 2 sets of experiments. The first, where you would take a nascent double-stranded DNA and radial label one of the strands. So you see that little dot up here, so we can probably say maybe this is P32. You're only labeling one of the strands. You're going to put this double-stranded, radial label double-stranded DNA into about 10 test tubes. Okay, so where we have an arrow, we might have a test tube. Okay, and within each of these test tubes, you're then going to add DNAs, which likes to cleave double-stranded DNA. And you carry out this reaction just long enough so that you get 1 nick in the double-stranded DNA in each test tube, okay? And because it's random, each of these test tubes will get a different nick, okay? So you're going to get different size fragments in each of the test tubes. You take all of those test tubes and combine them together, and hopefully you'll get an array of fragments that have been chopped up by the DNAs. Okay, so you'll take all those fragments and then run them on the gel, and it kind of looks like a ladder as depicted here, and you can detect them because of the P32. And remember, the larger fragments are at the top, and your smaller fragments are at the bottom of the gel. So that will identify how many fragments you carried out were generated from this DNA's treatment, from this double-stranded DNA. The second set of experiments is the same thing, same principle. You put your double-stranded, radial label double-stranded DNA into 10 different test tubes. You're going to add in your DNAs for a certain amount of time to make sure you get a particular nick somewhere in the DNA. But you're also going to add your binding protein, your DNA binding protein. So in this case, it's the RNA polymerase, which is RNAP. If the RNA polymerase binds to a specific region on the DNA, it will prevent the DNAs from carrying out a cut at that particular region. Okay, so you notice there's no arrows between this section, between here and here. So potentially, if there were to be nicks, those different size fragments will be absent. Okay, so you can pool all your 10 test tubes, you get a fewer number of fragments being generated, and you run them on the gel, and you should actually see a region where you don't have any fragments. And these would correspond to these different size fragments. Now, if you're cutting the DNA at single nucleotide sequence, you can actually figure out the sequence of your entire double-stranded DNA. So this will allow us to figure out where that RNA polymerase is binding on the double-stranded DNA. Okay, so this is how we identified promoter regions. And from this process, in analyzing thousands or hundreds of genes from prokaryotes, and I've listed a couple of them on this slide, there were 2 specific regions in each of these genes that the RNA polymerase was binding and was protected from DNA's treatment. So hopefully everybody's heard of the -10 and -35 region. Does this sound familiar to everybody? How many have heard this? Yeah? Okay. So the 2 regions and where RNA polymerase binds is the -10 region. It's called -10 because it's upstream from the start site of initiation, which is denoted here +1. Okay? So upstream means going this way of the initiation site. Downstream, so downstream, and this is upstream. Downstream would be to the right of the initiation site. Okay, so we see some similarities in this region. This was discovered by David Pribnow, so sometimes it's called the Pribnow box. And then there's another -35 region. Again, becomes a bit more variable over here. But if you take an average of these sequences, and like how many times each of these deoxynucleotides appear, you can obtain a consensus sequence. So a consensus sequence is an average. So an average of whatever deoxynucleotides we see. And you can see from here, the Pribnow box, its consensus sequence is TA, TA, AT, okay? I definitely want you to know this sequence. It's going to become important, so it's not hard to memorize, TA, TA, AT. And then the -35, again, you don't have to know this one. The distance between these 2 regions is about 11 to 15 base pairs on average, okay? And these numbers down here represent the frequency. So about 80% of the time, you'll have a T in that position. Look at that, 95% there's going to be an A. Okay, so this is just the percentage or the frequency that you will encounter that specific deoxynucleotide in the sequence. The -10 plus the -35 constitutes the core promoter region. Okay, and I've listed down here, so it's approximately around 40 nucleotides in length, so approximately. Initiation sites, typically, again, the percentages of frequency of the deoxynucleotide that's present. Most times it's a C because RNA synthesis typically starts with a purine, so most times A or G. Okay, so when we talk about generating transcripts. All right, any questions about these promoter regions that have been deduced using footprinting? Okay, so the consensus sequence is the average of these hundreds of sequences that were found through footprinting. Okay, so how does the sigma subunit, which I mentioned is the one that really recognizes the promoter regions, carry out that identification? And again, it's much similar to what we talked about in the first part of the course. It basically relies on the functional groups from a particular helix that project outwards interacting with these consensus sequences that have been identified. So if we look at the sigma subunit, its structure is depicted here. What are all these here? What type of secondary structure is that? It's an alpha helix, right? There's a key helix, it's called the recognition helix. That one is responsible for making hydrogen bonds using the functional groups from these amino acids depicted here. Again, you don't have to know them and the numbers. Transiently to identify the correct TA-AT sequence, okay? And again, if we have these genes, you're gonna make these interactions with this specific promoter region, all right? So again, make transient hydrogen bonds until you get the correct ones. So these are H bonds using the functional groups from these amino acids. All right? So amino acids are going to become important as we start moving through transcription and then eventually through translation. Okay, so that's the recognition helix that will, as the RNA polymerase is moving along the double-stranded DNA, it will test to identify correct hydrogen bond formation between these promoter regions. And once it's established, then transcription can begin. Okay, so I just want to get the terminology out of the way, because when we talk about upstream, downstream, I want everybody to be on the same page for the nomenclature. So if we think about our double-stranded DNA here, that's going to be used to carry out transcription, we have a 5' end and a 3' end. That's the non-template strand. Sometimes it's also called the sense. The 3' end to the 5' is the template strand, or antisense. And this is what the RNA polymerase uses to carry out transcription. So you use the template strand. Template strand is 3' to 5', okay? So if this is our RNA polymerase molecule, or protein, here's the transcript that's being synthesized, okay? So the nascent RNA transcript. And you can see, this is the 5' end, this is the 3' end. So synthesis, again, is 5' to 3'. All right? That transcript, in particular if it's an mRNA, you will use how many of these ribonucleotides to be able to translate into a protein? How many do we use to actually specify a particular amino acid? 3, right? 3 are in a codon, so for this one here, this is our first codon, so 3 of them. Does anybody know what amino acid is specified by AUG? Methionine, right. So your first amino acid, when you translate it, would be methionine. So met. Okay, and then you will use the next 3, incorporate your next amino acid, and generate your particular protein. Proteins, similar to what we see with the transcripts, are synthesized from the N terminal to the C terminal. Okay, so we go from N to C in terms of synthesis. As we're transcribing this, we read the transcript 5' to 3' and carry out the synthesis of our protein from N terminal to C terminal. The RNA transcript is complementary to the template strand in terms of sequence, okay? It is identical to what we see in the non-template strand, except for what? What's the major difference in the transcript? Yeah, where we have a T, we now have a U. Okay, so this is the major difference between those sequences. Okay, so a bit about the promoter efficacy. Typically, genes that have promoters that strongly resemble the -10 and the -35 consensus sequences are called strong promoters. And if they have a strong promoter, it means they're frequently transcribed. Okay, so I'll give an example here, sometimes 2 seconds, every 2 seconds in E. coli. If it has a weak promoter, so there's multiple substitutions in that consensus sequence for the -10 and -35, you might have maybe 1 every 10 minutes having that gene being transcribed, so 1 time. Typically, the distance between the -10 and -35 is optimal if there's about 17 nucleotides between them, okay? And these promoter regions are actually regulated in terms of how strong these promoters will be for the RNA polymerase by regulatory proteins. So you can have proteins that bind to them that can increase the efficacy of the RNA polymerase transcription. And then finally, for most of the genes, there's also another additional promoter region. It's called the UP element, and it provides increased stability for the alpha subunit. So it provides more contact for the alpha subunit to increase the stability of RNA polymerase binding. So we have three components, the -10, the -35, and then this UP element, which is found upstream of the core promoter region. I just have a slide depicting what a UP element looks like. And again, here's our start site, +1. Note that there's no 0, so we don't go +1, 0, and then -1, okay? So it goes +1 to -1. We have our Pribno box, our -35. Upstream of this is the UP element. There's really no consensus sequence to it. It just happens to be AT rich, okay? And it's about 40 to 60 nucleotides upstream. It binds, again, the alpha subunit at the C terminal of that particular subunit, and it increases the efficacy of our efficiency of transcription. It's not highly conserved, and again, just lots of ATs in that particular region. Okay, so we got the -10, -35, and this UP element. All right, so it's not the only promoter sequences or consensus sequences that we find in prokaryotes, that -10 and that -35. There are a couple of other promoter sequences that are specific to a certain set of genes. And these genes are typically expressed under changing environmental conditions. So where we have listed up here, the standard promoter for most genes includes that TATA box, like -10 region, and the -35. And that's recognized by the sigma 70. So you see the number there, 70. Okay, so that recognizes the standard promoter consensus sequences. As I mentioned, there are other sigma subunits that will recognize different promoter regions associated with a certain set of genes. So for the sigma 32, it actually recognizes these two -10 and -35 consensus sequences. That is the promoter region for heat shock proteins. What are heat shock proteins? What do they do? When would you need a heat shock protein to be synthesized? Yeah, so heat shock proteins tend to be chaperones. They help with what? What do they help with? Protein folding, right? So if you get an abrupt change in temperature, as I've listed here, the E. coli will actually synthesize lots of sigma 32, which will then recognize this specific -10 and -35 region that's associated with the heat shock genes. And then you'll start to get an increase in the transcription of those proteins. Likewise, there's a sigma 54 that is responsible for recognizing the nitrogen starvation promoters. And this is associated with genes that will be synthesized when the E. coli needs to process other nitrogen sources for energy. Okay, but the core one that will recognize that standard promoter is the sigma 70 subunit. But there are others within E. coli. And again, remember the sigma subunit is essential for recognizing the promoter region. I have a crystal structure of the RNA polymerase in the bound state with the double-stranded DNA. Here is the core enzyme in blue, and then we have the sigma subunit in the orange, okay? So you can see that the -10 element, this is where specific interactions are occurring with the sigma subunit, along with the -35 element. And again, these transient hydrogen bonds are made as it slides along the double-stranded DNA. Once it recognizes the correct ones, it will start the transcription initiation process. Okay, so it's essential for recognizing those two elements. It's a pretty cool structure. Okay, so how do we start transcription? You need the holo enzyme. Again, so it's a pentameric structure, making those hydrogen bonds transiently from their functional groups of a particular amino acid onto the double-stranded DNA. The process is very rapid, so the RNA polymerase latches on, it just randomly walks along the DNA strand, slides along, and then tries to identify those promoter regions. The rate of binding is about 10 to the 10 molar to -1, second to -1, and this is 100 times greater than if it was jumping off or on the double-stranded DNA. So kind of like DNA polymerase. So on or off of the double-stranded DNA. So it just latches on, tries to find those promoter regions, and then start initiation. And again, remember I told you once the sigma subunit finds those key standard promoter regions, it will help synthesize about 8 to 10 nucleotides, ribonucleotides of the transcript, and then it will fall off. It will just be released. And it can go off then and attach to another core enzyme. So in that sense, it's said to be catalytic. So then it will search for another core enzyme to start transcription elsewhere. Okay? So sometimes the sigma subunit is often referred to as a catalytic subunit. Okay, so let's describe the process of transcription. First, we're going to have our double-stranded DNA. We're going to have our RNA polymerase. This is the hollow enzyme. It will bind to the double-stranded DNA, find that specific promoter region through the transient hydrogen bond bonds that it makes through those key amino acids. And once it does find it, it will pause there. And this is called the closed promoter complex. Okay, once it identifies that key promoter region. At this point, transcription is still reversible. Okay, so this is a key step here. When it starts to unwind the double-stranded DNA at that promoter region, transcription becomes irreversible. Okay, at the open promoter complex. So we go from the closed promoter to the open promoter complex. And as I mentioned, typically the first ribonucleotide is a purine that's incorporated into the catalytic site to carry out initiation. Okay, so we get RNA initiation with a purine. So it can be an A, most of the times it's an A. So I have a question here. Which would have the largest KD? Okay, KD meaning dissociation constant, the hollow enzyme or the core enzyme of RNA polymerase. So I'll give you about 15 seconds to think about that. The largest KD. Anybody want to guess? A or B? Yes. Which one? How many say B? How many say A? Yeah, A is the answer. Why is A the answer? And I'll give you the difference. It's about 10 to the 5-fold difference. Remember, KD is a measure of the dissociation constant. So if you have a large KD, it means the affinity is high or low. So it has less affinity. So the hollow enzyme, I told you, has less affinity than the core enzyme. Okay? Everybody clear on that? You try to think of it in terms of Ka values or pKa values. Remember, if you have high pKa, remember it's low affinity, low pKa, high affinity of your enzyme. Okay, so we've started initiation. How do we elongate the chain? Basically, it's just a repetitive cycle of inserting ribonucleotides and creating that phosphodiester link. So we usually have that purine come in, we'll have our ribonucleotides around, and we start incorporating subsequent ribonucleotides. Once we get to about, here it says 4, but typically on average, maybe around 8 to 10 ribonucleotides in length, the sigma subunit leaves. Okay, so it falls off. You can go off and find another core promoter, core enzyme to associate with. And then the RNA polymerase has cleared that promoter region. And you just have the elongation process, which is just repetitive insertions of ribonucleotides. Okay? Some information about that core open promoter complex. If we look down into the molecular level, you can actually see that the unwinding of the double-stranded DNA constitutes about 17 base pairs in length, or an equivalent of 1.6 turns of the DNA. And here's our RNA polymerase that's moving along and incorporating the ribonucleotides. Again, the transcript exits the channel, so there's a channel within the RNA polymerase where our growing nascent chain will come out of. If it's an mRNA, will start being translated immediately. Okay, so here's another question. Which of the following DNA segments most likely represents the sequence where the transcript bubble begins to form during transcription initiation? So where we're going to unwind the double-stranded DNA, and explain your choice. Is it going to be A or B? How many say A? Okay, how many say B? Why are we choosing A? What's the difference between A and B in terms of sequence? Yeah, so there's lots of ATs here, which makes it easier to unwind than something that has a lot of GC base pairing, right? And if we look at our sequences for the -10 and the -35, well, the -10 is TA, TA, AT. So lots of TAs and ATs in that promoter region. So it's easier to unwind. Again, the process, just a reminder, we synthesize from the 5' end. So this is our triphosphate moiety over here at the 5' end. Usually we have that ribonucleotide being a purine, an A or a G. We have our incoming ribonucleotide that's incorporated using that 3' hydroxyl. So if you want to write 3' here. And the release of pyrophosphate, which drives the reaction forward, and we elongate our chains, okay? So the chains are formed de novo, so without a primer, and they grow in the 5' direction to the 3' direction. So keep those key features in mind. And another thing I want to introduce when we talk about the transcription bubble, where we've actually unwinded the double-stranded DNA, we have our RNA polymerase. As we unwind along the 3', 5' template strand, we are rewinding along the front end of the RNA polymerase. So unwinding here, rewinding the double-stranded DNA. Within the RNA polymerase, we have a new structure. It's called the RNA-DNA hybrid helix, okay? And it encompasses the template strand and partial component of the nascent RNA transcript. So here's our RNA transcript that's coming out. And here is our 3' hydroxyl at this end that will attack the incoming ribonucleotide to carry out that fundamental reaction. And remember, the RNA polymerase is moving this way. As this is rotating, where we're unwinding and rewinding, the RNA-DNA hybrid helix also rotates as well. And it rotates so that it positions that 3' hydroxyl correctly in the active site so that catalysis can happen. This structure is about 8 base pairs in length, okay, or 1 turn of the double helix. So about half the size of the actual transcription bubble, or the open promoter complex. And again, it moves very slowly, incorporating about 50 nucleotides per second. One key thing, and again, you notice there's a channel where this transcript exits. This channel constitutes key secondary structures, a helix loop helix that we see here, and a beta sheet. And there's a key phenylalanine residue, so if you just want to write phenylalanine here, that actually inserts itself into that RNA-DNA hybrid helix and forces the separation of the DNA from the RNA, such that that RNA can then exit through the channel. So it exits through a channel. Okay? So just know that there's a key residue, so that aromatic ring gets inserted into that RNA-DNA hybrid helix, and we get separation of the two strands, so that the nascent RNA chain can exit through the channel. Okay, so I think we'll take a 5-minute break here and come back and talk about the elongation process, which has been defined as a nucleotide addition cycle, as we just keep adding ribonucleotides over and over again. Okay, so take a 5-minute break, and we'll come back.